Have you ever stopped to think about how we get a better potato or, a banana that's more resistant to disease? It sounds pretty straightforward, but a lot of our favorite foods are actually clones. Yep, genetically identical copies. And that presents this really fascinating challenge. How do you improve a plant that's basically designed to be a perfect copy? It's a genetic puzzle that scientists have been tackling for decades. So let's dive in. Right here, you're looking at some of the world's most important clonal crops. Potatoes, sweet potatoes, bananas, they're all propagated asexually. What that means is a piece of the parent plant, like a tuber or a stem cutting, is just used to grow a new, completely identical plant. So if they're all just copies, how in the world do breeders introduce new traits like better flavor or the toughness to survive climate change? So in this explainer, we're going to unpack this whole process. We'll start with the basics. What exactly is a clone? Then we'll step into a breeder's shoes to really get their biggest genetic challenges. We'll look at the clever ways they actually break the cloning cycle to make improvements, explore the seriously high-tech tools they're using today, and finally, check out the future of food, a future where farmers are becoming key partners in the science. Okay, so to get started, we really need to nail down the fundamentals. What does it actually mean for a crop to be a clone? Here's the most important part. Clonal propagation is asexual reproduction. Now, think about it. Sexual reproduction with two parents shuffles the genetic deck every time. But this process, it's more like a photocopy machine. It just cranks out a genetically uniform population from just one single individual. Every plant is an exact copy. And this happens in a bunch of different ways. For potatoes and yams, we plant the tubers. For a crop like cassava, it's stem cuttings. Bananas and plantains grow from these things called suckers that pop up from the base of the parent plant. And in modern labs, you can even grow tiny little plantlets from tissue culture. Each method has the same goal, making sure the new plant is a perfect replica of the last one. So, this genetic uniformity is fantastic for farmers, right? They want a predictable, reliable crop. But for a plant breeder who's trying to make things better, it's a massive puzzle. Let's dig into the genetic dilemma they're up against. Okay, so let's break down this breeder's dilemma. On the left, with seed crops like corn or rice, the goal is usually genetic simplicity. You want to create these pure lines where the gene pairs are identical. But on the right, for something like a potato, it's the total opposite. These plants are naturally heterozygous, which means they have this rich, complex mix of different gene versions. You can think of it like a genetic treasure chest that's locked. The breeder's job isn't to empty it out, but to find the right key to unlock the best traits, all while dealing with extra complications, like having multiple sets of chromosomes, a condition called polyploidy. And that genetic complexity leads to a huge roadblock, something called inbreeding depression. To put it simply, these plants are naturally wired to cross with others. If you try to force them to cross with themselves or with close relatives, the results are, well, pretty disastrous. The offspring are often weak, sickly, and just don't grow well. It's a fundamental problem that forces breeders to get really, really creative. So, how do they do it? The solution actually seems kinda counterintuitive. To improve a clone, you first have to stop cloning it. Breeders have to temporarily hit pause on that cycle of asexual reproduction. This slide lays out the conventional strategy perfectly. It all starts by bringing in sexual reproduction. Breeders very carefully select two parent plants with traits they want to combine. Then they cross them to create actual seeds. And this is where the magic happens. It creates a ton of new genetic variation. They'll grow out thousands of these unique seedlings, pick out the absolute best individual plants, and then only then do they go back to cloning, making countless copies of that new superstar for testing and eventually release. So that's the classic approach. But modern science has an entire advanced toolkit to totally supercharge this process. Let's look at some of the powerful techniques that let breeders get results that, not too long ago, would have seemed like science fiction. This quote really gets to the heart of a powerful strategy called analytical breeding. The keywords here are ploidy manipulations. It's all about using the plant's chromosome sets as a tool to unlock genetic diversity, especially from wild relatives that might be holding the secret to, say, disease resistance or drought tolerance. So what exactly is ploidy manipulation? Well, it's basically intentionally changing the number of chromosome sets in a plant. A great example is the bananas we eat. They're triploid. They have 3 sets of chromosomes, which is what makes them sterile and seedless. Breeders create them by doing some clever chromosome math. They cross a parent with 2 sets, a diploid, with a parent that has 4 sets, a tetraploid. The result is a unique hybrid with those traits that we all love. Another really fascinating tool actually comes from the lab. When you grow plants in tissue culture, little genetic changes can just spontaneously happen. This is called somoclonal variation. Now, this can be a bit of a double-edged sword. On one hand, it can be this surprise source of awesome new traits, but on the other, it can introduce changes you don't want. So breeders have to manage it really carefully to either take advantage of it or make sure it doesn't mess things up. To generate a massive amount of genetic diversity really quickly, breeders use a cool technique called a polycross nursery. Basically, they plant a whole group of elite parent clones together and just let them openly cross-pollinate with each other. It's like a genetic lottery that creates thousands of unique offspring, giving breeders this huge population to search through to find the next superstar clone. And we really can't overstate how important tissue culture is, because diseases get passed directly from parent to clone. Viruses can just build up over generations. But growing plants from their meristem, that's the very tip of their growing point, in a sterile lab environment, acts like a reset button. It creates perfectly clean, virus-free little plants. This is absolutely critical for producing healthy crops and even for rescuing valuable plant lines that have gotten infected. So we've seen the science, but the story doesn't end in the lab. Not even close. The future of breeding is becoming more and more collaborative, bringing the scientists and the people who actually use the plants, the farmers, together. Traditionally, we had what's called formal plant breeding. This is where scientists worked on research stations to create modern varieties that were designed to work just about everywhere. But that top-down approach didn't always work for farmers in specific, challenging environments. The shift now is towards participatory plant breeding. This is a collaboration where the selection process happens right there in the farmer's fields, making sure the new varieties are actually adapted to local conditions and what people actually want. And this is such a crucial point. In many parts of the world, farmer-bred varieties, which have been developed over centuries of on-the-ground selection, still outperform the modern ones. It just highlights the immense value of local knowledge. When breeding becomes a demand-driven process, with farmers as key decision-makers, the adoption rates for new varieties just skyrocket. So why does all of this complex science and collaboration really matter? Well, because improving these crops is absolutely central to addressing some of the biggest challenges we're facing as a planet. The ultimate goal here is to breed crops that can stand up to the pressures of a changing world. Plants that can grow in harsher climates, that can fight off new pests and diseases, and that can sustainably feed a growing population. This isn't just some abstract science. This work is right on the front lines of global food security. And all this leaves us with a final and pretty critical question. The power to engineer these elite clones is immense. But in our search for the perfect plant, how do we balance that pursuit of perfection with the need to protect the broad genetic diversity that is really our best insurance policy against the unknown challenges of the future?